Hello, grade 12s. I hope you're having a great evening. I'm not having the best evening myself. I'm having a bit of an allergic reaction right now. So if you see some red spots in my hands, don't be alarmed. Alright, so anyways, yesterday we discussed about somatic cells and the organization of genetic information within those cells. Today we're going to discuss chromosome numbers and karyotypes, which is how we examine chromosomes. Alright, so let's begin. The number of individual chromosomes each cell contains varies from one species to another. Human somatic cells have 46 chromosomes. This can be organized into 22 pairs of autosomes, which are homologous chromosomes, and one pair of sex chromosomes, which may or may not be homologous. Autosomes are labeled from numbers 1 to 22, and sex chromosomes are labeled X or Y. The sex chromosomes are what determine the sex of a person. A human female has 2 chromosomes labeled XX. Human males has 1 X chromosome and 1 Y chromosome. Alright, let's take a look at what homologous chromosomes mean. Homologous chromosomes are similar looking, but they are not necessarily identical. They carry the same genes, which are the areas of DNA that contain specific genetic information at the same location, or a more scientific term would be locus. The reason homologous chromosomes are not identical is because they can carry different forms of a gene called alleles, which are represented by the letters in this diagram. We will discuss more about alleles next week. A cell that contains a pair of homologous chromosomes is said to be diploid. The diploid number in humans is 46, or 23 pairs. A cell that contains unpaired chromosomes is said to be haploid. Human gametes, which are the reproductive cells, i.e. eggs and sperm, are haploid. The haploid number of chromosomes in a species is designated as n equals 23. A diploid cell has 2 N chromosomes, which equals 46 in humans. Some organisms are polyploid, which means they have sets of more than 2 homologous chromosomes, such as 4N, 8N, 3N, etc. The N number varies among species. So what I want you guys to do is I want you to search online to find different species with different N numbers. And be prepared to share your findings with the class tomorrow. Alright, so let's take a break. Pause the movie if you wish. Grab a cup of coffee, grab a tea, whatever. And then we'll continue on to our next discussion. Alright, hello again. So now we're going to move on to our next discussion. So we are going to look at how we as scientists can examine chromosomes by creating a karyotype. But before we do that, first we're going to look at some of the characteristics of a chromosome and the properties that are shared among homologous chromosomes. So autosomes 1 to 22 and the sex chromosomes are distinct from one another in many different ways, as you can see here. For example, chromosomes in an organism can vary in the overall length. They can have different locations of their centromeres, and the staining properties, which is this distinct pattern of banding when a chromosome is stained. However, these 3 characteristics are the same in chromosomes of homologous pairs, which you can see here. So therefore, scientists can use these characteristics to identify individual chromosomes and to match pairs of homologous chromosomes, as you can see in this diagram here. So what you're seeing here, this is called a karyotype. And the particular set of chromosomes that an individual possesses is called an individual's karyotype. So when you're looking at this, can you tell me, is this a female or male? Think about this. What do you see? Alright, so I noticed here that there's only one X, but even though it says one X, look at these two chromosomes. Are they homologous or are they not homologous? Alright, so this particular karyotype is quite boring. It's just black and white, so you can't really see the different staining properties. So let's take a look in color, shall we? Yes, that is much better. You can see the different staining properties much clearer. Alright, so to prepare a karyotype, scientists collect a cell sample and use chemicals to stop the cell cycle when the condensed chromosomes are most clearly visible, which is right before cell division, which you can see here as I have indicated with green. So then the scientists, they stain the cells to help them identify the individual chromosomes. These chromosomes are photographed and they complete the karyotype by organizing the images into series of homologous pairs, as you can see here. Alright, so that's it for today. Remember to re-watch any parts of the video if you were ever confused with any materials covered today. And also remember, don't forget to look at the summary information held on the website. And also don't forget to look online for different species with different chromosome numbers, different from humans, so you can share that with the class tomorrow. And don't forget to participate in all the activities for today's lesson. And I hope you have a wonderful rest of your evening, and I will see you all tomorrow. Bye-bye.